So these are three characteristics of the genetic code. The fact that the genetic code is universal means that we can move pieces of DNA from one organism into another organism, and when we move them across and we do transcription, then that foreign piece of DNA will be transcribed and translated into the same protein as it ordinarily would be in the naturally occurring species. So universal means that we've got the same genetic code. For example, okay, if we've got UUG, whether we're a bacteria or whether we're a human cell, we're still going to put into position at the ribosome leucine. So the genetic code is universal. The genetic code is said to be non-overlapping, and we'll dig a little bit deeper in the understanding of this in a moment. Degenerative means that in some cases, and again, if we look at this threonine, all of these codons have got different base sequences, but all of them code for threonine. So it's said to be degenerate. So in some cases, and not in all, there are multiple codons which code for the same particular amino acid. So the universality, the non-overlapping nature of the code, and the degenerative nature of the code is three important characteristics. Okay, we met this already, this idea that we've got a so-called start codon. The start codon is always AUG. That's important. And we've also got some parts of the messenger RNA that are referred to as a stop codon, and the stop codon stops translation. So we need to think now of the mechanics and the sequence of events that takes place in translation. So what we need to appreciate is that the first thing that happens is that the small and the large subunit sandwiches between it this messenger RNA strand, and that messenger RNA strand is pulled along until we hit a start codon. Remember that we met this idea that the transfer RNA has got that cloverleaf structure. We've got the anticodon here, and the anticodon is going to be complementary to the codon sequence. I want us to think of the ribosomal structure in this sense, and when you make your own notes, I would recommend that you use a structure which is like this. So we've got what's called an E site, a P site, and an A site. So at the start of translation, what happens is that the messenger RNA is sandwiched between the large and the small subunit. That's pulled along until we get to a start codon AUG, and that start codon will sit within the so-called P site. What then happens is that we've got a transfer RNA molecule, which has got the anticodon sequence UAC coming into position and temporarily forming hydrogen bonds between the codon and the anticodon. This brings into position the first of the amino acid. So this is called the P site. What will then happen is a second transfer RNA will come into position into the A site. The A site is the so-called acceptor site, and when that transfer RNA sits in here, again, we're going to have complementary base pairing between the anticodon and the codon, and it will bring a second amino acid into position. That means that we can have a reaction taking place, a condensation reaction taking place between this amino acid and the amino acid which will sit next to it. So this is called the P site, or the peptidyl site, because this is the position when this P and A site is occupied. This is where the peptide bond forms. When the peptide bond does form, what happens is that this bond between the transfer RNA and the actual amino acid is broken. The whole thing shifts along by 3 codons, and we're going to shift these 3 codons here, and then we're going to bring 1, 2, 3, these 3 codons into the position of the A site. So we're moving it in a non-overlapping manner into the next sequence, and we're going to carry on that process. This first transfer RNA will go into the E site, which will then exit. And again, the McGraw film shows you this in some detail, and it shows you animation-wise, which allows it to be more easily understood. So if we look at this diagram here, again, we can see much the same thing. You've got the idea of the anticodon and the codon. So here we've got the P site, and as you can see here, the P site allows the peptide bond to form when the A site is also occupied. When we form that bond, this is broken, and you can see what's happening here. There's no amino acid, it's already been bonded. So this amino acid here was once upon a time attached to this particular transfer RNA molecule. And you can see also that we're looking at it at 3, and then we've got the next three, and the next three. We're not overlapping. We're not saying CGU and then saying GUA. We're pulling it along each time in terms of triplets. It's useful when you're making your notes to discuss this in terms of 3 stages. We've got the initiation stage, the elongation stage, and the termination stage. What you can put before the initiation stage also is details, and we will come to this in 7.2 in a little bit more detail about the activation of transfer RNA. So the activation of transfer RNA means that the correct amino acid is attached to the correct transfer RNA molecule. That's an energetic process. It requires ATP. Then we've got initiation. We've got the ribosome and the messenger RNA coming together. We've got the ribosome moving the transfer RNA along until we hit that start codon. It moves in the 5' to 3' direction. We've got the anticodons on the transfer RNA molecule aligning, and we've got that opposite the complementary base pairs of the codon. So again, this is a summary. You can read it for yourself. The elongation process tips the hat to this idea that in order to form a peptide bond, you require both energy and you need an enzyme, and the enzyme is associated with the ribosome itself. Termination is when you hit a stop codon. These next slides, okay, itemize things. They've got a nice diagram here, and what this will do is it will tell you in sequence 1, 2, 3, 4 the different stages, and this might help you. Look at the next one. This might help you to sequence your notes together. It does come up as a past paper question quite frequently, so it's worthwhile having a series of diagrams with a series of prose written alongside it. I also suggest that you articulate this out loud so that you can be very, very fluent in terms of the description of the process of translation. We will add to this when we look at 7.3 a little bit more detail than we've met just here. So if you look at these syllabus statements here, it's saying that we've got to be able to use and apply the genetic code. So this link here is very, very well worth exploring. When you're looking at any kind of tabular information, just take note of the fact, is this table showing you the codon sequence? It should be, but they may give you the anticodon and expect you to work backwards in some senses. Similarly, got the same sort of idea here. You do not need to learn all the names of the amino acids at all, okay? But if you want to know what those shorthand stands for, they are here. And this next slide shows you some examples of the kind of thing that have come up in terms of past paper questions. And if you work through the PowerPoint slide, which is within the Google Classroom, you can see that we've got the answers which are written up there for you. It also reminds you of a few things to look at. Finally, we've got this syllabus statement here about the production of human insulin by genetically modified bacteria. We did meet this at GCSE. We'll touch on it again very briefly here, but what we will do when we come back to Unit 3 is we look at this again in a little bit more detail, and we add to the idea of genetic modification of organisms. But essentially what you are doing is you're cutting out the human insulin gene. You're using an enzyme called restriction enzyme, which cuts open that particular gene. You then take the human gene and you also need to use a prokaryote. So the prokaryote, remember, has got small circular pieces of DNA plasmid. You take out the plasmid from the microorganism and you cut it, again using the same restriction enzyme. And that same restriction enzyme means that the sticky ends are the same, which means we can insert the human gene, which you can see here, into the plasmid. This is then referred to as recombinant DNA. Recombinant DNA is DNA which is made up of DNA from more than one organism, and in this case it's more than one species. And then we take that recombinant DNA, we place it back into the bacteria. So this bacteria is then referred to as a transformed bacteria, a genetically modified bacteria, a transgenic bacteria, all the same thing. And this particular prokaryote has got the human gene in it, which, because the code is universal, means that transcription and translation of that human gene can take place, and this prokaryote is going to make human insulin. You don't want to have one of these bacteria. You want to asexually reproduce this bacteria, so you've got lots and lots and lots of them, and all of these will be built up within a fermenter, and that fermenter will allow you to have many, many microorganisms all producing human insulin, which is very easy to purify. So the genetic modification links to this idea of the genetic code being universal. This wouldn't happen if it wasn't for the universal nature of the code. This one here reminds you about restriction enzymes. We come back to this later on, but restriction enzymes are, as you can see here, are sometimes looked at as biological scissors. They're going to look, all enzymes, remember, are specific. So the active site of this enzyme is going to look for a very, very, very specific base sequence here, and it's going to cut the gene here, and similarly there, to allow the human insulin gene to be isolated. The same restriction enzyme means we've got the same sticky ends. Sticky ends means we've got these unpaired bases, okay, and that means that it's easy to put this one and this one together and for hydrogen bonds to take place. We are going to need to use ligase, and ligase allows the phosphodiester bond to be formed. So this we come back to later on. This should be reasonably familiar from GCSE, and I can field questions in the lesson if required.